Hello dear students, welcome back to Sajjan biology classes. In today's session, we are going to discuss about the restriction enzymes. In general case, the bacteriophages, it affects the bacteria. But in the year of 1963, the scientists came to know that there are two enzymes that were restricting the growth of bacteriophage within the bacteria. And later they isolated. And they came to know that one is adding methyl groups to the DNA. That is nothing but a methylation of DNA anta kareeತೀವಿ. By adding methyl groups to the DNA, what it happens then? So actually it silences the retroviral elements. And another enzyme, so actually that was cuts the DNA. And these two enzymes later they called as restriction endonucleases. And the first time the restriction endonucleases were discovered by a scientist called Hamilton Smith. And which is the first enzyme is isolated then? You can see here that the first restriction endonucleases which is isolated called HIN2. Okay. And among totally 900 restriction endonucleases were isolated from the 2 and 230 strains. And you can see I have written here 6 base pairs. Means when you look into a long polynucleotide chain of DNA is comprising of many base pairs. And in that, particularly the restriction enzymes identify the site and it cuts. And those sites are called recognition sites. And HIN2, it identifies and recognizes the site and it cuts 6 base pairs. And there are different set of enzymes and which cuts the particular number of nucleotide base pairs. And another example here that ALU1. Arthrobacter luteus 1, which cuts the DNA at 4 base pairs. Okay, and let us discuss that the restriction endonucleases are particularly having certain principles. Means the naming of restriction endonucleases, it is based on certain principles. Let us discuss that which are those the principles. You can see this is what the first letter which is referred as a genus. Okay. And which is written in capital letter. And next second 2 letter refers to the species which is written in small letter. And next one is that the next letter that represents the strain. And the next one you can see this is what Roman numbers. And you can see here Roman numbers indicate the order in which the enzyme were isolated. And let us discuss that what are the types of restriction endonucleases are there and what is the function of these enzymes. Here restriction enzymes belongs to the larger classes of enzymes called nucleases. And here there are 2 kinds of enzymes are called exonucleases and endonucleases. Just you see this is what the double stranded DNA. And what they will do this exo and endonucleases. Exonucleases which cleaves at the end of DNA, and where that endonucleases they cleave in between the DNA. That's it. Okay. And next we are discussing in detail about this restriction enzyme, how actually it acts on DNA. And just we have taken example here that ECOR1. So is an enzyme which is acting on both vector on foreign DNA by recognizing a particular sequence called GAATTC. Okay. And once after the recognizing the recognition sequence, it leave one nucleotide base pair in both vector and as well as foreign DNA. And by this way, it forms the sticky ends. And these sticky ends are joined by the ligase enzyme. And then it forms the recombinant DNA. The one more important thing we need to note here that restriction endonucleases enzymes, they identify the sequence in palindrome order. So what are these palindrome sequences? Any sequence when you identifies means the restriction endonucleases when it identifies the sequence in the DNA which will be same in both the direction. You can see this is the forward direction and this is what the backward direction. So GAA-TTC in the forward reaction, and in the backward you can see the same GAA-TTC, GAA-TTC. So, and such sequence can be called as an palindromic sequences. Even there are many examples that have given in the textbook to understand Malayalam. So when you read from forward or when you read backward, it gives the same meaning, Malayalam. So that is what. And even you can give many examples like that, madam. So madam, M-A-D-A-M, M-A-D-A-M. So this kind of sequences, so these are the examples, and particular example belongs to this restriction enzyme is that echo R1 which cuts the DNA, GAA-TTC, GAA-TTC. The palindromic sequences are the sequence where the base pairs will be the same in both strands. Okay, so whenever you read from forward as well as the backward, the base pair sequence will be the same. And these kind of sequences are called in palindromic sequences ways to call. I hope you understood, and still do you have any doubts regarding this? You can drop your doubts or comments in a comment section. I will reply for your queries. Please like, share and subscribe. Thank you.